All right, good afternoon. We should get started. Welcome to the afternoon session. My name is Chang Zhou. I'm a faculty member in the MCV department here at UC Berkeley. And my laboratory studies HIV transcription control and latency. The first speaker of this afternoon session is Professor Peter Sarnow from Stanford University School of Medicine. And the title is Modulation of Hepatitis C virus and Insig 1 by specialized microRNA 122 complex. Thanks.
Thank you. And first, I want to really thank Lauren for setting up this incredible meeting. And so many people came and such great science so far. I think this is really great. Thanks for doing that. And hopefully others will step up and at UCSF and Stanford, and we can make this an annual event. This is really cool. Okay, I like to, as you just heard, I like to talk to you about hepatitis C virus and a microRNA 122 that is subverted by the virus. Before I go into this, I just want to tell you a couple of facts about microRNAs. And you probably know there are 800 microRNA genes in the mammalian genome. Each microRNA is predicted to regulate about 10 to 100 target mRNAs. So if you do your math, come to the conclusion that a third or fourth of all mRNA in a cell are regulated. Importantly, and as you will see, several microRNA are conserved among different organisms, from plants to invertebrates to vertebrates, and some are expressed in a tissue-specific manner, and I will show you some striking examples. Now, we got into thinking about microRNA when we thought, how do they work? We know they bind to 3' non-coding region, but nobody really knows how they mechanistically work. How do they downregulate gene expression? So we thought what we should do, we should find out a cell culture system where we have a microRNA expressed in one cell but not in another cell. It should be highly abundant, and maybe then we pick out a normal endogenous target mRNA and study the interaction. And so we came across microRNA 122 because it's an RNA that is majorly abundant in the liver. In a normal liver, like in your liver, you have about 66,000 copies per cell. In transformed liver, the numbers of copies drops to about 15,000. So we thought, well, is this true? So we did a northern blot analysis, and indeed, in human and in rat liver, there's a whopping amount of microRNA 122, not in HeLa cells, it's on non-liver cells, as you know, and in cultured transformed liver cells, again, as predicted, in lower abundance. And we noticed that it was not expressed in HepG2 cells, which are also liver cells. Now, at the same time, we have been working for a long time with hepatitis C virus, specifically with the iris element, and we couldn't really not notice that we know certain genotypes can replicate and grow in HUH7 cells, but not in HepG2 cells. So we were wondering, actually, is a coincidence, or does microRNA 122 maybe interact with the HCV genome and enhances its replication? And we heard this morning from Melanie Ott, great introduction about the life cycle of hepatitis C virus. I just want to mention it's 10,000 nucleotide positive stranded RNA genome. The 5' end, it has an iris, an internal ribosome entry site that grabs directly ribosomes. At the very 5' end is a very conserved stem loop structure. The 3' end doesn't have a poly A tail, it has a conserved RNA structure. Now, when we looked, we found actually at the 5' end, just between the stem loop 1 and the iris, there were two sites to which microRNA 122 could potentially bind. And they could bind like this. So in green, you see the microRNA. In blue, you see mRNA sequences, nucleotide sequences in HCV mRNA. What is known, nucleotide 2 through 7 in a microRNA needs to interact in perfect Watson-Crick base pair interaction with the target mRNA. So then you have a very bona fide microRNA binding site. The other thing we know, that these sequences shown here in blue were absolutely conserved in the gene bank in all HCV genotypes which you can find in the gene bank. So that was very important. And sequences such as here, they were diverse, but the spacing between the two sites was actually very conserved, 50 nucleotides. Now, so we thought, how do we test whether this is functionally important? So we thought we should inactivate the microRNA. So how do you inactivate a microRNA? So in the early days, people used siRNAs, and that doesn't really work because a microRNA is also incorporated in RISC. Of course, as a siRNA that should destroy it. And then we learned over the years certain antisense oligonucleotide antisense RNAs can actually bind and sequester microRNAs in cells and in organisms. And I want to show you this piece of data because I was so excited when I heard Ron Plastock in 2005 in Cold Spring Harbor at the meeting giving a talk about locked nucleic acids. Locked nucleic acids, these are nucleic acids where the 2' and the 4' position is linked by an oxymethylane group. Now these oligonucleotides result in a very stable interaction with their target microRNA. And in this case, he used it in situ hybridization to identify microRNAs in zebrafish. MicroRNA 206 is expressed in the muscle. This is the muscle of the zebrafish. You have one mutation in the locked nucleic acid oligomer. You see very little to no signal anymore. MicroRNA 124 is only expressed in the brain. Indeed, this is the brain of the zebrafish. And then he said, this is microRNA 122. He says it should be expressed in the liver. He says, I didn't know that zebrafish has a liver, but I guess this must be the liver of zebrafish. And I thought he was joking. So when I came back from the meeting, I went to a textbook, anatomy textbook. And indeed, this guy was right. This is the liver of zebrafish, and microRNA 122 is in there. So after his talk, there was a poster session. I went to the poster session, and there was a poster by a company, Xantaris in Denmark. And they used LNAs. They actually have the patent on LNAs to really set up microRNA arrays. And they could show that these LNAs were perfect, really terrific to do microRNA arrays. And indeed, microRNA 122 was on there, and it really seems to capture and sequester microRNA very well. And I asked him, tell me more about LNAs, and he says they are stable. If you have phosphorothioate on them, they go straight into the cell. They actually, if you put them in the bloodstream of an animal, they go straight into the liver. And my jaw just dropped, and I said something like, can I have it? And he said, sure. So we got LNAs against 122, and we added them to cultured cells infected with hepatitis C virus, and HCV RNA was gone. 80% was gone. So then we wondered, well, does the microRNA bind directly to HCV, as our hypothesis was, or does microRNA 122 binds to another mRNA that's important for viral growth? So what we needed to do, make a mutation in the HCV sequence to which the microRNA binds at site 1 or site 2 and see what effects are. Indeed, if you change the C to a G, what you see is HCV RNA abundance is gone. So the endogenous microRNA, the wild type one, cannot function. However, if you ectopically express, and we express duplex mimetics, then you can restore function. This is genetic, not biochemical. Genetic evidence, microRNA interacts with site 1. How about site 2? Same thing, you make a mutation, you lose it, you can restore with a microRNA mimetic that has a mutation to bind to the mutated HCV site. Do they need to bind at the same time? Well, you make a mutation here and here that you can rescue independently, and indeed only if you have both mutated microRNA mimetics there, you have rescue of HCV. So this tells us that HCV binds two microRNA to the same molecule at the same time. There's an offshoot possibility that one microRNA binds, does a hit, and then runs, and the second RNA binds, but this invokes more mechanistic explanations. And a simple explanation is both bind at the same time. So this is our model. And then the next question was, well, what is the mechanism? Why does it enhance HCV RNA abundance? So we spent a lot of time, and we found it has little effect on translation. It has little effect on RNA replication. So we thought, what about RNA turnover? So we tested RNA turnover, and I want to show you one series. We have several series of experiments. One series of experiment, and it will show you that when you really do kind of a very seemingly boring experiment, but carried out by two really expert people, you can really get a lot of mileage out of this. And the seemingly boring experiment was, how about we make mutations, two nucleotides at the time in the microRNA, and see what effects it has on HCV, because we really wanted to know what are the roles of these end sequences here. So Erica Macklin and Selena Sagan did this experiment, and as you can see, you mutated these here, you lose HCV RNA abundance. These you can mutate. They have some effect, but they don't really kill HCV RNA. 15 and 16, to our surprise, these two Gs at both sides here or here, sites 1 is red, sites 2 is blue, completely killed HCV. The 3' end, again, was more variable. Now, when you get a negative result, we thought maybe the 15-16 mimetic is dead. It doesn't work. Maybe the wrong RNA gets into the risk. What's going on? But it's not dead, because if we take this microRNA mimetic 15 and 16 and test it in an siRNA assay, and we tested it also in a microRNA assay, indeed what we find, it's very functional. So there are clearly important for hepatitis C virus. Then we noticed that nucleotides 1 through 4 are also very conserved in the gene bank. So we had the idea that 15 and 16 maybe interact with the very 5' end of the HCV genome. Then you would have the 3' end of HCV sticking out. And then maybe these sequences and these mutations in them are tolerated. So we tested this again. Just focus on the red bars, please. We made a mutation. Changed the C27 to A, and we changed these 2 nucleotides to 2 Gs. We transfect this RNA, no RNA abundance. You killed RNA abundance totally. Now, if you add in a microRNA mimetic, where you have a change of a G to a U that could base pair with the A, and a C at C and a C at 15 and 16 that could base pair with nucleotides 2 and 3, which are now Gs, you rescue RNA abundance. So we think what we have is a very complex RNA molecule where 2 microRNAs bind in tandem, a conserved space, or the same nucleotides loop out. And interestingly, the end nucleotides really stick out of at the 5' end of HCV. If you change this U to a DT, a deoxynucleotide, you kill HCV RNA abundance, but you don't affect siRNA activity, you don't affect microRNA activity. If you take away 2 nucleotides here at the 3' end, you completely abolish HCV RNA abundance, but you don't affect RNAi and microRNA activity. Something is very, very special about this. So what is this? We think, from what does microRNA 1 to 2 could really protect HCV? We think innate immune responses. We are virologists. So what does this mean? Well, we need to know, how does the end look like of HCV? If it has a monophosphate, there are nucleases, such as exon 1, they could degrade it. If it has a triphosphate, well, maybe RIG-I gets activated. It gets activated by triphosphate-containing RNA, which you normally don't find in cells. Who knows? Maybe it has a capping activity. Of course, we know it doesn't have a capping-predicted polymerase such as dengue, which we heard of this morning. But who knows? We know that the nuclear capping enzyme can function in a cytoplasm, and who knows? Maybe it's capped. So we have been spending a lot of time figuring out what does the end look like, and many people ask me, so what's wrong with you? Why don't you know yet? But it's very, very difficult. It's not that simple, and we're working very hard on it. But maybe it's true that there may be different end moieties at different times during the life cycle. We really don't know yet. Now, one thing was really strange. We know that the microRNA 1 to 2 is in great abundance in the cell over HCV. So in a normal liver, 66,000 microRNA molecules per cell and about 15,000 HCV RNA. But we know, for example, we know that microRNA 1 to 2 is transcriptionally regulated. Every night is a nocturnal mechanism. Every night, transcription goes up in animals, and in the morning, transcription shuts off. So you make tremendous amount of microRNA 1 to 2 on a daily basis, but the turnover of microRNA 1 to 2 is 3 weeks. So there's something really weird going on. So we were wondering, does a specialized microRNA 1 to 2 associate with HCV, the old one, a new one? And we wanted to explore this in more detail. And indeed, what we find when we overexpress a microRNA mimetic, we actually can enhance HCV RNA abundance. Although the endogenous one is already there in abundance over HCV, if you stick more into the cytoplasm, HCV RNA abundance goes up. So we thought, well, maybe is it the delivery to the cytoplasm, or is this kind of mimicking what the newly transcribed RNA might do in the cell? And that could be, for example, an RNA being synthesized when the light goes off at night. And so we're wondering whether a transcription of 1 to 2 has an effect on HCV RNA abundance. And we found this paper last year, which came out from a group by Young et al. And they found that compound 3, shown here, actually can enhance transcription of microRNA 1 to 2, but not that of microRNA 21, shown here, the mature microRNA and the primary microRNA. So Cecilia Sarnow asked the question, well, what happens if we add compound 3? And she could show, indeed, microRNA 1 to 2 abundance went up. And if you focus on the boxed lanes here, you find, indeed, in another analysis, if you add compound 3, we know microRNA 1 to 2 goes up, HCV RNA goes up. What about proteins? Same thing. This is a control with DMSO. If you add compound 3, HCV core protein goes up. So we know newly delivered or transcribed microRNA enhances HCV RNA abundance. So the question is, so what's the fate of the microRNA in the cell, of old one and new one? So what we did an experiment where we simply run a sucrose gradient shown here. We collect the sucrose gradient at A254 nanometer, and what you see here, ribosomes, 40, 60, 80S. These are mRNA associated with 2 and more ribosomes. We call them polysomes. Now, if you look what happened to microRNA 1 to 2, I should say we add cyclohexamides to freeze the ribosome so they don't run off, they don't fall off. Well, you find it at the top of the gradient and you find it at the bottom. So dogma so far has it some microRNAs bind to translating mRNAs and control translation. Here's microRNA 21. You see a similar pattern, although more is at the top. Here's actin mRNA. What we find is when we infect cells with HCV, the RNA, the microRNA 1 to 2 is located less so at the top, but it's really located at the bottom of the gradient where we think are polysomes, mRNAs, and ribosomes. That does not happen with microRNA 1 to 2. So the question was, are these mRNA molecules which are attached to ribosomes to which microRNA 1 to 2 gets attached? So what do? Well, get rid of the ribosomes. And the way we get rid of a ribosome is we add puromycin. Puromycin is an elongation inhibitor, and it dissociates ribosomes. So you end up actin, for example, migrates towards the top of the gradient. And what we find, indeed, microRNA 21, it's a little hard to see, also goes to the top of the gradient. And microRNA 1 to 2 ends up in a new position in fractions 9 to 12 in the gradient. So these are microRNA complexes probably devoid of mRNAs and of ribosomes. It's maybe an aggregate or something really strange. So we were wondering whether this complex is really EDTA sensitive, because EDTA also takes away ribosomes. Ribosomes need magnesium in order to be held together. And when you do this in uninfected cells, you see there is a fraction 9 to 11 where microRNA 1 to 2, you find microRNA 1 to 2, again stripped of ribosomes. But in HCV infected cells, the same fraction persists. It's still there. microRNA 21 doesn't do this. Now, we think that this is a complex of microRNA 1 to 2, which is normally in the cell, which HCV absorbs. It grabs it to do something which we really don't know. And if 1 to 2 is not there, something horrible happens to HCV and it gets degraded. When you heard Melanie Ott's talk this morning, she talked about core and lipid droplets. So we have some evidence that hepatitis C virus might be in this fraction, and indeed there is some overlap of hepatitis C virus RNA, and we find that core, that's the protein she talked about, which associates with lipid droplets, is also in this fraction. Not so much NS5A, which is a replication protein. So we have the hypothesis, and we test this more, and we have not probed these plots for DGAT1, but we will, whether maybe microRNA 1 to 2 protects HCV at a stage where it's really sensitive, where it is kind of an Achilles heel for antiviral interaction, and that intervention, and that might be from being translated to being packaged, but we don't know yet for sure. So if you start to begin dreaming about microRNA 1 to 2, and of course, can you use it as an antiviral target, you need to know what's the normal function of 1 to 2. Well, 2 papers came out at the end of 2005 where they used oligonucleotides, this anti-1 to 2 oligonucleotides. These were not LNAs, but subsequently Santaris has repeated all of these results with this experiment, getting the same results with LNAs. If you sequester 1 to 2 in a mouse liver, nothing happens. The mouse is fine, the liver is fine, it's not stressed. Eventually, you kill the mouse, take the liver, you do a microarray. And what you find is what everybody finds. 300 genes go up and 300 genes go down. And what you can do is you can ask the question, is there a pathway? Is there a pathway that's in common? And indeed, some of you might know this, these are all genes involved in cholesterol biosynthesis. So the normal function of 1 to 2 is to upregulate cholesterol biosynthesis. Indeed, animal studies and phase 1 clinical trials have shown when you sequester 1 to 2 in the liver, your plasma cholesterol levels actually goes down, and also your liver cholesterol levels goes down in animals, of course, we don't know in human. So we were wondering, what is the target of 1 to 2? And so we were looking for a negative regulator of cholesterol biosynthesis, right? That should be target, so cholesterol synthesis goes up. And you look, we call it the shiny pebble syndrome. So you look at many candidate genes and you test them and they turn out all to be wrong. So we came across something that was very interesting, and this is the work now I'm talking about of Kara Norman. We came across something that when you read this paper, overexpression of Insig-1 in the livers of transgenic mice that inhibits SRABP processing and reduces insulin-stimulated lipogenesis. And you know that SRABP is a major transcription factor that regulates cholesterol synthesis. So you know that this is an interesting negative regulator of cholesterol biosynthesis. When we read this paper, we noted that one figure in this paper had exactly the same lists the same genes as the one in this paper. So this tells us that microRNA 122 most likely regulates Insig-1. So before I go into the data and show you evidence for this, I briefly want to tell you how is cholesterol levels, how do you regulate it, how do you regulate cholesterol levels in the cell? It's complicated. I try to make it very simple. High cholesterol levels, if you have high cholesterol levels, Insig-1 is being produced. It binds to HMGCR. It's a rate-limiting enzyme in cholesterol synthesis, and it degrades it. More importantly, it binds via SCAP to SRABP and retains it in the ER. Remember, that's a transcription factor that turns on all these genes. Now, low cholesterol levels come, so you want to get rid of Insig. Basically, Insig gets degraded by the proteasome. SRABP gets processed, goes out into the nucleus, and kicks on genes. So when we looked at Insig-1, we were gratified to see indeed it has predicted binding sites for microRNA 122. And we discovered that there are 3 isoforms. The isoforms only differ in the 3' non-coding regions. And interestingly, one of it, the one giving rise to a 1.4 kilobase mRNA, has a site that overlaps, has a binding site for microRNA 122 that overlaps with the cleavage and polyadenylation signal. So what happens is normally Pol2 goes along here and sees an AUU or an AAA, UAA signal, that's a polyadenylation signal, and 30 nucleotides further down, this protein complex cleaves the mRNA, and then you polyadenylate. So 122 seems to interact here. That was very curious to us. So we needed to set up a system in which we can study whether Insig-1 is regulated by microRNA 122. So Kara did this in tissue culture cells by setting up a system where she starved overnight. So now we have low cholesterol levels here, and we add back mevalonate and sterols, and then suddenly cholesterol levels go up, and Insig should go up. And indeed, Insig protein goes up, and we see it more strikingly when we add a proteasome inhibitor. And next question was, what about the 3 isoforms of Insig mRNA? And what we find indeed, what we found to our surprise, that all 3 mRNA, they go down. At this point, we have higher cholesterol levels, so we expect that some mRNA to be up because we need to translate Insig protein. But what we find in polysome gradient, that this mRNA, the 1.4, although it's in lower abundance than at low cholesterol levels, it's actually very efficiently, very quantitatively associated with polysomes. So this is the mRNA that synthesizes Insig-1 protein. Well, what about what is microRNA 122 doing? So we take LNAs and we sequester microRNA 122, and we find the ratio of this Insig isoform mRNAs changes. So specifically, if you look here, if you sequester 122, the 1.4 goes up and the 1.9 goes down. So what this means is our hypothesis that the 1.4 is downregulated. Remember, this is you sequester, it goes up. So normally 122 downregulates the 1.4 isoform. And we think that mechanism involves polyadenylation and the cleavage site of the 1.4 kb Insig isoform mRNA. So finally, I want to very briefly in 2 slides tell you where do we go with these LNAs. And I told you that people, the Santaris in particular, have shown that indeed if you take LNAs against microRNA 122, you put it in PBS, you put it in the bloodstream of a mouse, and we have shown with them in a chimpanzee, what you find is plasma cholesterol levels go down. We do not have an animal model for hepatitis C virus. That's the biggest nemesis in our field. We are really handicapped. We don't even know where HCV comes from. We have no clue where it comes from. We know HIV and retroviruses have been around 10 million years. We only know from human liver samples that HCV probably was in the human liver for 10,000 years. That's all we know. We do know we can infect monkeys, chimpanzees, and establish a chronic infection. So what Santaris did together with Robert Lanford at the Regional Primate Center in Texas, they took a monkey that was chronically infected with HCV. This monkey is fine. It has been infected with the virus. They don't develop liver cancer, and has been infected for 12 years. So what they did is they just focused on the left panel. They gave this monkey a high dose, 5 mg per kilogram, and a lower dose for a weekly treatment for 10 weeks of LNAs against microRNA 1 to 2. And what they find, indeed, the cholesterol levels went down. And what you see here is the HCV titer. It went down by 2.5 logs. Then they stopped giving the monkey an LNA, and what they find is it still persisted, but then eventually it came back up. Now the question was, of course we know I didn't mention this, we know that from pharmacokinetic studies, when you stop treating an animal with LNAs, 7 weeks later, the LNA gets secreted via the kidney. So this happens here 7 weeks later, right here, week 17, the LNA gets secreted, it's out of the system, HCV comes back. I was absolutely convinced these are all revertant viruses that have a mutation in the binding site for 1 to 2 and simply grab another microRNA. So here we go. But then these guys did deep sequencing and they found not a single mutation was found in any of these viruses. So this is very promising. But we heard this morning, maybe, I'm not saying we should treat infected patients with LNA for 43 weeks. That's the standard of care treatment because, of course, there are 300 other targets and some are proto-oncogenes. But what we heard this morning, maybe a rapid drop of a virus by one reagent and together maybe with another reagent, right, might really help or give the innate and adaptive immune system a boost to really combat the virus. So indeed, Santaris has announced September 10 last September that they are in phase 2 clinical trials. And these are actually, they treat people who were not on standard of care. So these people have never were never treated with interferon. And they do that in San Diego and in a couple of countries in Europe. So in summary, I told you that HCV has a little helper in liver cells. There's a really strange microRNA complex, and that's the one HCV has subverted. And it downregulates Insig 1, an mRNA isoform by a mechanism by which normally microRNAs do. And I told you that in the chimpanzee model so far, there's good evidence that it lowers the yield of virus. And I mentioned some of these people. That's the most important slide. I forgot to mention Teresa Abraham and Cara Parker. They did the sucrose gradient and the microRNA complex analysis. And I mentioned Erica and Selena and Cecilia and Cara, and Catherine Chopling was the first to start these studies. And of course, I want to thank my collaborators over the years, Joachim Ellman, Zachary Kampinen. And thank you for your attention.
So I'm curious about your observation that you think that that even though this microRNA is expressed really highly in cells, that if you add more of it, you can still get a downregulation. Do you think that is there any evidence that HCV is binding a pre-processed form of it, a primicroRNA or a pre-microRNA, and that's how you're getting selectivity for newly transcribed versions of it, or? Right.
The answer is we really don't know. But indeed, there are more and more evidence comes out that precursor microRNA can have function. The question is, what is the stability of the precursor microRNA in the cytoplasm? How fast does it get processed? We really don't know, but this is, you're right on the money. It's a very distinct possibilities. That why the newly transcribed one is so important. You don't know.
Peter, back here. So is there any evidence that miR-122 binding affects the localization of the viral RNA in some way? And also, can you just comment on, you didn't go into it explicitly, but you think that XRN1 degradation is not part of what is happening with miR-122, and maybe it's protection of the end of the RNA, and what your evidence is for that?
Right. So localization studies, we have done that. We can look for 1 to 2, and it's in distinct patterns in the cell in HCV. Maybe there is a little overlap, but clearly the majority of HCV cannot be, by this kind of assay, cannot be detected by immune fluorescence to overlap. But I don't know it's whether the detection system is not good enough, and we can only see 1 to 2 when it's in high abundance in one compartment. We really don't know right now, but yeah, and there's clearly a lot of 1 to 2 that doesn't interact with HCV, and that might be, we don't see any distinct pattern. And the XRN1, there is one study which has been reported a long time ago, that in replicon cells, maybe there is a population of HCV that is sensitive to XRN1, but we don't see this as clearly when we infect cells with the real virus. So the verdict is still open. I'm open to the possibility maybe there might be a transition, there might be, maybe if there's a triphosphate, it might get converted to a monophosphate, or we don't know.
Question in back.
Sorry. Oh, it's very fascinating. In your second slide, you showed HeLa with no miR-122 and Hep2.
Right.
I'll just comment. Walter Nelson Reese, who's an alumnus of Berkeley, passed away. He had an expose in Science some years ago showing that Hep2 is really HeLa. So you confirmed him. But my question, I have a more basic question. Do you have evidence that there really is binding of miR-122 to HCV RNA? I didn't, maybe you showed it, I didn't see it.
Correct. No, it's the answer. It's all genetic. We have evidence for genetic interaction. We have no evidence so far that the microRNA physically binds. We don't have an in vitro biochemical assay yet. So we are trying to, tag the microRNA and see whether we can cross-link. But no, you're right. It's just genetics.
The last question.
So I just have a quick question regarding the miRNA. Have you looked at some of the statins to see if they actually increased production of miR-122?
Say again. Have you used statins to inhibit the cholesterol pathway and see if you get upregulation of miR-122 instead of using LNA? We have not done that, but Frank Cesari has done it, and that was a great excitement when we had these results. And of course, many patients are on statins for other reasons. The answer is no. We have no evidence for it.